All right, thanks for the introduction, and very happy to be here. So to complete the introduction, I'm at Kalev VIB, and yeah, mostly wet lab, but also some computational. And yeah, we'll give an introduction on the technologies that you're maybe working on. So from single-cell RNA-seq, but also from long before, we know that there's a large complexity of cell types in our bodies, and we not only know that they're all there, they also have, most of them have a very specific location in the tissue, and they have specific relationships to one another. So the whole field here is basically how to measure where certain cell types are, where certain genes are expressed, etc. And all these methods do that by doing some sort of spatial measurement. There's very different flavors, many different flavors. We will go over them. To get a sort of an atlas or an understanding of what is where. This is an example of cell types. It's not necessarily bound to cell types. It could also be many other modalities that you might be interested in, like a single gene or a pathway, etc. So the goal of this lecture is really to understand the technology behind all these black boxes that many of you are using. So we're going to look a bit at the chemistry that's behind, the instrumentation that's behind all these methods. And you might think like, well, I'm a bioinformatician, I don't care, I just want my data set. Why do I need to learn about what's happening in this black box? But I think it's very important for you to know so that you can choose the right technology for your question, because they all have advantages and disadvantages. You need to know the limitations and the biases of these methods, and also to recognize technical artifacts, because that can also happen that you think you're looking at a biological signal, but it might actually be a technical artifact. So we'll go over the main approaches. I've grouped them into 4 groups. There is a lot more, but we don't have the time and to cover all of them. And we will be comparing these 4 main approaches based on their resolution, the sensitivity, the number of targets they can measure, and the area that they can cover in a single assay. The methods can be split into 2 major groups. The first group is based on microscopy, and the second group is based on sequencing, which is actually also microscopy, but then outsourced to Illumina, unless you're doing nanopore. The microscopy methods, they mostly use some sort of in situ hybridization method, where a probe is hybridized to a complementary sequence of RNA or cDNA. The microscopy methods fall apart in 2 subcategories. One is the barcoded fish approaches that we will talk about, and the next one is the in situ sequencing methods. Then on the other side, the sequencing approaches, they use spatial barcodes, and they also fall into 2 different groups. One where the RNA moves towards the spatial barcodes, and then the second group, spatial tagging, where the spatial barcode moves towards a target. We can also split these 4 main categories in a different way. The barcoded fish methods are targeted, so you need to know beforehand which genes or which targets you want to see. Then the others, the middle to the in situ sequencing, can be targeted, can be untargeted. Nowadays it's mostly targeted. The spatial sequencing is generally untargeted, but there are some flavors where they are targeted. But usually transcriptome-wide then, and the spatial tagging is generally untargeted. All right, I made a list here of most of the methods that fall into these categories. Okay, let's go over them. We will start with barcoded fish. So as I said, these methods are all based, or most of them are based on in situ hybridization, and more specifically the fluorescent version of that, which is called fluorescent in situ hybridization, or FISH, where you have your RNA target, in some cases a cDNA target, and then you design a probe. You need to do this beforehand with a complementary sequence that will bind to the RNA, and then you can hang a fluorescent molecule onto this probe so that you can later visualize it. Yeah. Then you also have a single molecule resolution, a variant of this, which is called single molecule FISH. where you can really count individual molecules of RNA. And this is a very powerful method and very sensitive, and because you can count individual molecules, so you get a really good quantification with high sensitivity. And the way this is usually done is that you design many probes, many complementary probes that target an RNA molecule and tile it completely. They all have their own fluorophore, or you have also some more complicated things we'll get on in a few slides. But they basically tile the whole RNA, and this generates a high concentration of fluorophores on that RNA molecule. And when you then put that under the microscope, you see these spots. So in this image, you see in blue the nuclei, and then all of these tiny spots are individual molecules of RNA, and the color corresponds to one of the 3 genes here. This is a very simple variant of this technology. So this is great, very quantitative, very nice, but there is a major limitation in microscopy, and that is that there is not so many distinguishable colors in a microscope. So if you have a typical microscope, you can maybe see 3 or 4 different fluorophores or colors in your setup. If you have a very special microscope, you can maybe go to 7, but that's about it. And as you know, the transcriptome, or we have about 20,000 genes in our genome, so 4 to 7 is really far off from what we would like to measure. This whole field, the imaging side of this whole field, really got kickstarted by the idea of performing multiple cycles of staining a tissue, then imaging a tissue, and then removing the labels again, so that you can then do another round of labeling. And the scaling was really introduced by the barcoding approach. So this approach of labeling, imaging, stripping, labeling, imaging, stripping, already increased the throughput, the number of genes a little bit, but then with the introduction of the barcodes, also the number of genes scaled a lot. So I made here an example of 3 different, where we used 3 different fluorophores in 2 different rounds to label 9 different transcripts from 9 different genes. This is what that would look like under a microscope. So we're going to hybridize in the first round all these different probes, and then we see all these dots. For instance, here we see 3 red dots, but at this point we don't know which gene they belong to. We know that they must come from RNA A, B, or C, but which of the 2 you don't know. So then you strip the probes off and you hybridize the probes of the second round, and now you see that these same molecules changed color or didn't change color. And if you then afterwards align all these images, you can see that these dots move, have a sequential color code. In this case, this molecule is red first and then green. And then we go back to our lookup table and we see red, green, that must be RNA B. So in that way, we can identify 9 targets in 2 rounds with just 3 different colors. When this was first published, we thought it would scale really well because the formula goes like this. The number of targets is equal to the number of fluorophores you have to the power of the number of rounds, which would mean that with 4 fluorophores in 8 rounds, you would be able to do the human transcriptome 3 times over. However, it was not as easy as that, unfortunately, basically because of 2 main limitations. One is called optical density. Our microscope have a resolution, which means that these dots have a well-defined size. They're actually smaller than what we see, but this is the limit. We hit the limit of the microscope, so the dot size is determined by the resolution of the microscope. And that means that if you stain more and more RNA molecules each round, at one point they will start to overlap, just because the molecules are physically closer to each other, but we can't resolve them in our imaging system. So here I drew some arrows. Like this is a big dot. It might be 1 molecule, it might be 2 molecules, might be 3. I don't know. And if you do more and more genes at the same time, you will run into these problems more and more. The second thing is that when you do many rounds of labeling, stripping, there's a lot of fluidics going on. There's movement of the sample, and you will accumulate errors over time. So that is a way that you would lose signal. For that, there is a solution which was introduced by Murfish, and this is using sparse barcodes with error correction. So the barcodes we saw before, they were staining each RNA molecule every cycle with a different color. Murfish introduced these sparse barcodes like this binary code here, which would mean that every digit here is a single cycle, and your RNA molecule would be stained in the first cycle, not in the second, not in the third, then again in the fourth, et cetera, et cetera. And in that way, you can also identify your RNA molecules, but you not stain all of them at the same time. So you solve a little bit this optical density issue. The other thing is that you can use special barcodes that have an error correction built in. I'm not going to go into all the details here, but if you make sure that your barcodes are unique enough so that many bits would need to flip to mutate one barcode into the next, you can build in sort of error robustness, and this is introduced by Murfish. So in this way, you can identify a lot of RNA molecules at the same time in the tissue. Here's an example of this data set. So every dot here is a single molecule of RNA. In this experiment, we did 440 genes. I'm just showing you 30 here. And you see there's like big cloud of RNA molecules, and it looks nice, but to actually analyze this, many methods go through cell segmentation. So that's the next step. So you would detect your RNA, then you would do a counter stain of your cells to sort of see where they are. You can make a segmentation mask, that's the white line here, and then you can count the number of molecules that's inside each cell. And then you can get your cell by gene matrix. Cell segmentation is a big challenge, and there is also many different ways that you can do it. It's also very tissue dependent. So here's a couple of examples of how people have been doing cell segmentation. You can stain your nuclei. This is the most simple or like most straightforward method. You can, if you label your nuclei, you can segment those. Usually they're a bit separated from each other. And then you could, as a proxy of the cell, you could maybe expand them a little bit. And this was done, for instance, in Visium in the early days, but now they have a different approach. If you're very lucky and your tissue allows it, you could use a membrane staining, but this sounds like the holy grail, and it works for very few tissues, but for those tissues that it works for, it can be very nice. Other approaches are to stain both the DNA, like in the first approach, but also stain the total mRNA to get sort of a proxy of the cell body. Once you have these segmentation masks, you can, like I said, count the molecules inside these masks and then compile the data into a gene by cell matrix. And this is for you bioinformaticians very important because the imaging data sets are usually very large. So they're multiple terabytes per data set, and now you get to the order of megabytes, maybe a gigabyte, which is much more workable. All right, I already said that segmentation is very challenging, and you also have a little bit of practice with it. I just highlighted in the images I showed you before some of the issues. For instance, here in the nuclei, there is this sort of vague blob. I don't know what those are. If it's one cell, two cells, I don't know. Also, the segmentation algorithm doesn't seem to really know. With the membrane staining here, it's a bit ambiguous what is happening here. Is this where the cell boundary is? In the last method, there is a huge difference between the total mRNA count between different cells. So some cells are very big, they have a lot of RNA, so they get really nice green cells here. But then you have a lot of other cells that are very small, they don't have much RNA, so they are missed with this kind of approach. That's all to say that segmentation is a real challenge. There's many limitations. So one is the counter stains used, like to stain membranes, nuclei, organelles, filling cells. These are a bit limited. Maybe the biggest problem is that there is unclear ground truth. Like we, as scientists, don't know where the boundary is, how we're going to teach an algorithm where the boundary is. And also there is a resolution issue. And I try to illustrate that here. This is electron microscopy data from the mouse brain. And you see very nicely 3 cell bodies. They're big. But in between the cell bodies, there's a lot of the protrusions. These are axon dendrites, other filaments of different cells, and they are very small. So when we zoom in a little bit, you can start to see them, that they're really, really small. And if we now overlay that with the highest resolution we get in regular wide field microscopy, you see that many of these features are smaller than the pixel size. So it's a fundamentally hard problem to perform proper cell segmentation. What many people do, of course, is that they only focus on the cell body as a workaround. But yeah, just to say that it's hard and it will stay hard, this problem. Fortunately, there is progress. People are working a lot on different counter stains. Also, algorithms are improving a lot, especially with computer vision, AI. Its segmentation is improving a lot. And also, Naveed will talk about this with segmentation-free approaches that circumvent this whole issue. All right, here's an example, a few examples of well-established methods in this segment of barcoded fish. We have SeqFish or SeqFish Plus that I just took one. There's many publications, but I just took one. Here they showed that they could do 10,000 plex decoding of 10,000 genes in situ, which is great. Merfish is maybe the most established method in this field and also the most famous, I think. And this is, I think, the same slide as Ahmed showed, where they really made a whole atlas of the mouse brain. Showing that it's a really robust and powerful method. And also they have a paper where they reached 10,000 genes. A few weeks ago, the people from Nanostring, now Bruker, had a very nice bioarchive where they showed that they could do the whole human protein coding transcriptome of almost 19,000 genes, which is a very impressive feat. Of course, if you're working with these data sets, the commercial platforms are way behind this. They do 500, maybe a few thousand max. But it's likely that it will get there. All right, so just to summarize this part, the barcoded fish, we have a couple of methods here. Murphy SeqFish are the most prominent. There's a bunch of commercial platforms that you probably are using if you're in this segment. The resolution is diffraction limited. It's determined by the numerical aperture of the microscope objective. They will probably not tell, the companies will probably not tell you that. They usually, or like many people, when they talk about resolution, they talk about the pixel size of their camera, but that's not the whole story. So just keep in mind that the resolution is somewhere in this range, and this depends on the color of the fluorophore that is used. So resolution is a more complex story than some of the companies try to make clear. In general, the detection efficiency for these methods is relatively high, really on the upper end. I'd put an asterisk there because, of course, if you're not imaging everything or if you're making some shortcuts, it could fall lower than this. So depends a bit on what happens inside the black box if they can actually reach this. The gene throughput is very good, from a couple hundred, I think, generally used to, it's proven now, it's theoretically possible to do the whole human transcriptome. When the commercial platforms will reach there, we still have to see. The spatial throughput is a bit limited. These methods require very high magnification imaging, which means that the field of view of a single image is very small. So to cover a large area, you need to do a lot of imaging. So in general, it's a few square millimeter to maybe a square centimeter, or maybe soon a few square centimeter. All right, within this segment of barcoded fish, there is also an amplified version. This originally came from the in situ sequencing group of methods that we will talk about later. but they now also apply a barcode rather than sequencing. And these methods rely on rolling circle amplification. This is a way to amplify a DNA construct, and it works quite well in situ. So what you do is that you either start from mRNA, which is modernly done, or before, when you first make cDNA of your RNA targets, and then you hybridize a padlock probe. It's called like that because it's sort of this padlock shape. You need to gap fill this middle, and then you can use enzymatic amplification to make many, many copies of this circle, and it will just go on. That's why it's called rolling circle amplification. Then, of course, you get many copies of the same probe, and if you then put a fluorophore labeled probe on there, you will get a very bright signal, or much brighter than compared to a single molecule fish. Just to give you an example of these methods, this is a HIBIS from the Mats Nilsson lab, where they use this to do barcoded detection. Maybe good to point out here is that they have a dense barcode, so there's no zeros in here, but I guess that Xenium is using a sparse barcode. So that's maybe good to know. These methods, HIBIS, HIBR-ISS, which is the direct RNA version of this method, their resolution is a bit limited, not necessarily by the microscope characteristics, although of course also, but also more about the amplicon size. So the amplicon size usually is 0.5 micron to 1 micron big, and this determines the resolution. The detection efficiency is a bit lower than fish, although still good, but this is because it needs to do many different enzymatic steps that are unfortunately not super efficient. The gene throughput is somewhere between 100 to 1,000, and the spatial throughput is very good because this signal is very amplified. You can image with lower magnification objectives, and you can more easily scan a larger area. So it's centimeter scale or even a couple of centimeters, square centimeters. Then let's go to the next group of methods, in situ sequencing. So like the name says, we're now doing the identification of the sequence in situ inside the tissue. Originally, they are also, all these methods are also based on rolling circle amplification, but then rather than doing a barcoded approach, they do a sequencing approach. And this is sequencing by ligation. So you have all your copies, then there is either a barcode that you, like a barcode sequence that you put into your padlock probe, or this could be the gap-filled portion here. So this could be the sequence of your RNA. And then the method uses ligation, so it hybridizes these probes with one determined nucleotide and then random nucleotides here. And then if you see that the red probe binds, you know that there must be a T at this position. Then you melt these probes off, you remove them, strip them off, and then you do a next cycle where you now have your determined nucleotide in the second position. And then after a while, you can reconstruct the nucleotide sequence. Like I said, this can be done in two ways. It can be either done to sequence a barcode to identify predefined targets, or you can do de novo sequencing. And in this, this is the first paper that introduced it from Mats Nilsson's group. They looked at different point mutations in different cells. The other group of methods in this section is StarMap, and they use a slightly different sequencing approach, a slightly different padlock design. They use serial sequencing, and they can also do that to map many genes over very large tissues. And this is another mouse brain atlas to show how robust this method is. And it's a nice data set. All right, so these methods, ISS and StarMap, are the academic publications. Xenium originally originates from this branch, but now moved to a barcoded approach. And then there's Stereo-omics. Resolution, again, is limited by the amplicon size and the characteristics of the microscope. The detection efficiency is the same as for the other RCA-based methods. Gene throughput is tens to thousands. I think it's a bit higher than this number that I put here. And also the spatial throughput is very good. All right, let's move on to the next section, the sequencing-based methods. Just to remind you, we had spatial transcriptomics, spatial sequencing, where the RNA moves, and then we had spatial tagging, where the barcode moves. First, let's talk about spatial transcriptomics, where the RNA moves. Yeah, this was kickstarted with a method called spatial transcriptomics, not to be confused with the whole field, where a microarray of spatially defined barcodes is used to capture RNA. So here is this is a glass slide. All these spots contain a spatial barcode. This is just a sequence of DNA that we know, and that we know is in this location, and then it has a poly-T sequence. Then a tissue is placed on top, and the mRNA, which has a poly-A tail, is led to diffuse out of the tissue onto the capture slide, and then they hybridize to the poly-T. When they do, you can remove your tissue, do RT, and prepare your sequencing library so that your reads will now have the spatial barcode plus a bit of the RNA so that you can identify the RNA molecule. This is a picture of the original publication where they applied this to the factory bulb of the mouse brain. So all these spots are a capture spot, and here they cluster them to show that they capture different regions of the tissue. This was quite quickly spun out of academia into a company called Spatial Transcriptomics, which was subsequently bought by 10x, which now named the platform Visium, which most of you are using. The original Visium had a spot size of 55 micron in diameter. Just to remind you, most cells, depends on the tissue, of course, are around 10 to 15 micron in diameter. So probably a spot contains more than one cell. For this issue, you have a lot of deconvolution algorithms now. And another thing to note is that there is spacing between the spots, so there's empty space between those spots. Visium is by far the most used method. And here I just highlight a famous paper where they applied it on the human brain, and they see the different layers of the cortex in the brain. I think this is a well-used data set that is used as a benchmark for many methods as well. But recently, last year, I think 10x announced a new version of Visium, which they call Visium HD, which has a much, much higher resolution of the array. So now the bins are continuous, so there's no gaps in between the different capture spots. And the smallest bin size is 2 by 2 micron, which they then computationally bin into 8 by 8 micron. So this is a subcellular microarray resolution. But the principle remains the same here, that the RNA would move and get captured there. Just to remind you, this resolution of the array is not necessarily the resolution that this method will give you, because RNA will diffuse also laterally before they get, diffuse is not all straight down. So diffuse also a bit laterally, so the imprint or the blot is not a one-to-one copy of the situation in the tissue. So there is a little bit of diffusion going on there. But this resolution is already so much better than what it was before. Here is an example of from 10x where they did the regular Visium and the HD on, I think, consecutive sections, and yeah, you really clearly see the increase in resolution here. I want to add to this is that 10x now also has a machine. It's called Site Assist. This is a method that was not first in an academic preprint or paper. So for us, it's a bit guesswork what actually happens in here. Most of the methods, I think I'm quite confident that we sort of know what happens inside these machines because we know the academic papers where all these companies are originating from. But of course, in the future, these companies will develop so fast to develop new technology, so it might be harder for us to really understand what's happening inside. And that's also the case with the CytOSys. So what we think is happening is that they turned Visium in a targeted assay where you have your tissue on the sample slide, you have your RNA. To those you hybridize probes, and then rather than the RNA diffusing to the capture slide, it's the probes diffusing. And I guess the advantage here is that these probes are much smaller than these large RNA molecules. They're uniform, which might give you a better diffusion and blotting characteristics than if you would rely on moving the RNA. Also, you can put multiple probes on the RNA. I'm not sure they do that, but you could put multiple probes on the RNA so that you have a higher chance of capturing that molecule. All right. More methods in this category are Stereo-seq. This relies on also an array, but this array is made a little bit differently. This is a large array with spots where on every spot there is they perform rolling circle amplification to amplify a spatial barcode. Then they sequence these spots in situ. So now they know where each spot is located and what their spatial barcode is. And then they attach a poly-T sequence, very similar to Visium, put the tissue on top, the RNA moves, gets captured, and is then sequenced. Because these spots are very small, they're 220 nanometer in diameter, with a sub-micron spacing, they also get a very high resolution with this method. Here's an example of one of their data sets, which I think is very beautiful. This is the cross-section of the mouse embryo. And they see all these different tissues. It looks quite wonderful. And then in this category, there is also a group of methods that use the microarrays that are made by Illumina. So Illumina makes these sequencing chips, which are in essence a microarray with a lot of different spots where you can capture your RNA molecules that they use to sequence your sequencing library. But these people from the method SeqScope, OpenST, and NovaST have used this principle or used this surface to also generate capture spots so that they can do spatial transcriptomics with this. Similar to the previous method, Stereo-seq, you also need to sequence your spatial barcodes before. So you take an Illumina chip, you sequence the first few bases of the barcodes that are on the surface so that you know which one is where, and then you take it apart, cut it into pieces, put your tissue on, and then let the RNA diffuse. And now, after the RT, you can now have your spatial barcode and your RNA identity in the same sequencing read. So then you need to do another sequencing. But this nicely uses these arrays that Illumina worked on very long and very hard to make them perfect. Okay, summarizing this section. So the methods are spatial transcriptomics or Visium, slide-seq, Stereo-seq, and the Illumina-based methods. The resolution is sort of determined by the spot size, going from 200 nanometer to, in history, a bit like 100 micrometer. But of course, the RNA diffuses, so this is not a full story. The detection efficiency is generally low, but it is full transcriptome, or anything that has a mRNA, poly-A tail will get captured on these slides. So gene throughput compared to the imaging-based methods is much better. Spatial throughput is also good. It's going from several square millimeters to even several square centimeters. All right, then the last bit, we'll do this very briefly, which is spatial tagging, where the barcode moves towards the target. There's a few different methods here. First one I want to highlight is dBit-seq. This is a microfluidic approach where you have this microfluidic device, and this has all these channels. These white blobs are basically like the wells where people put in a row or a column barcode, and they then get flown through these microchannels and across this bridge in the middle. This is where you would put your tissue. And you see that they have two chips, one horizontal for the rows and one vertical for the columns. And when you then do hybridize your barcodes to the target, after the first flow, you see all these lanes, the rows that are barcoded, and then they flip the chip, switch the chip, and then they barcode the columns, and you get a grid pattern, and then you know where your sequence, and then you do sequencing, sorry, and then you know from which row-column combination your targets come from. It's important to note here that the tissue in between the channels where it's black, they are lost, they're not used. So you are only looking at these square tiles. The resolution is somewhere between 10 and 100 micron, depending on the channel width. But the very amazing thing about this technology is that it was really easily adaptable to any of the existing single cell methods. So here's an example of some data where they sequenced all the RNA. They had proteins with a barcode that they could sequence, so they could sequence the spatial location of proteins. Here it's a histone marker that they could sequence. They could do ChIP-seq, all in situ. So this was a great platform to bring in existing technologies into and give it a spatial dimension. All right, then the last two methods. This one is called slide tags. We would have a barcoded beads. These are the same beads that are used for the microfluidic single cell approaches, where these barcodes are released from those beads and would diffuse into the tissue, tag a nucleus or cell, and then they proceed with single cell RNA or single nucleus RNA-seq. And now they know the spatial location of their single cells, and then they can place them back into space. So you have the advantages of the existing pipelines of single cell RNA-seq, but now they also give it the spatial coordinates. Of course, important to note is that not every cell that is tagged will also be sequenced. So there is a bit of a loss in your tissue. If you're familiar with this structure, the hippocampus, you might notice that there should be more cells in this layer here. Similar idea here is with a method called SciSpace. They have a microarray of spots instead of beads, where also they diffuse into the cells, they tag the cells, and then they proceed with single cell RNA-seq. Spots are 73 micron. There's some gap in between, and here they notice that they measured 2.2% of the nuclei that were originally in the section that also ended up in the data set. But again, because it uses existing single cell pipelines, it's easily adaptable and will give you extra information. So these methods, they have a resolution that is determined a bit by the spot size. For the last, there is also a bit of diffusion going on, but it's somewhere in this range. The detection efficiency is on the lower end or similar to single cell RNA-seq. The gene throughput is full transcriptome, and then for DBIT-seq and maybe also for the others, they can also use many other technologies, so it's not only limited to transcriptomics. The spatial throughput can be, differs a lot between these different methods that I mentioned, but it's from several square millimeter to a square centimeter. But they would be possible to scale. If you want further reading, there's this great website linked to this paper. The paper is quite hard to read if you're not already, it's a review paper, it's quite hard to read if you're not already fully into this field, but they have an amazing website where they review basically the whole field from chemistry to bioinformatics, and I would really suggest reading that or looking certain bits up if you need. And that's it. Thank you.